The following protocol is intended to show the isolation of primary astrocytes from brains of pup rats to perform the oxygen glucose deprivation, or OGD. Exposure of purified astrocytes to an oxygen and glucose deprived environment is a model that allows us to test their reaction to some of the conditions found in ischemic stroke. Morphological changes after exposure to oxygen glucose deprivation can be observed and measured using selective astrocytes markers such as GFAP. In summary, these isolated cells exposed to ischemic-like conditions can be used to test new pathways activated in hypoxic conditions and novel drugs that protect against ischemic stroke.
In this first part of the protocol, astrocytes will be isolated from pup rat cortices. The purpose of this procedure is to obtain a primary astrocytic cell culture to perform oxygen glucose deprivation experiments. The materials required for this procedure are the following: sterile flask for cell culture, sterile 60 mm Petri dishes, 1 operating scissor, 1 microdissecting scissor, 1 curved forceps, 3 microdissecting tweezers, 2 microdissecting forceps slightly curved, 1 microdissecting forceps angled. Cell sieve mesh number 60 and number 100. 2 beakers. 70% ethanol spray.